Biology, Part 11. Organization of protein molecules. The primary structure of a protein is defined as the sequence of amino acids in its molecule. The sequence is vital because through predictable interactions between different parts of the protein chain, it determines the three-dimensional shape of the protein molecule. This shape is obviously important in enzymes where the configuration determines whether the molecule on which the enzyme acts, that is, subtracts, will be able to fit in. Therefore, whether or not the enzyme will work. The secondary structure is also very important, for it refers to protein as regular arrangements of polypeptide chains. And then there are X-ray crystallographs. X-rays are scattered by the crystals from the characteristics pattern, and they move accordingly. There are different shapes, and there are different predictions for ionizations as well. Denaturation and renaturation. Most proteins lose their biological activity and are said to become denaturated in unfavorable conditions of temperature and pH. Denaturation can occur in temperatures 50 to 70 degrees Celsius. It does not normally affect the covalently bonded backbone of the protein or any of its disulfide linkage, but the protein or any of its weaker hydrogen bonds or electrovalent bonds are broken so that the polypeptide change onward, forming loose random coils. The complex shape of protein is lost and it's no longer functional correctly. While there are enzymes and metabolism, as we talk about them, the mechanism of enzyme action. At any moment, hundreds of chemical reactions are in progress in a typical living cell. These reactions provide energy and help to maintain supply of many different substances needed for growth and repair. The remarkable fact that all these reactions contribute to an organized overall metabolism is due to almost entirely the production of special substance called enzymes. The purpose of this one is to outline the nature and properties of these enzymes, to explain their role in regulating the metabolism. All enzymes are globular proteins, and within the cell they have two fundamental roles. Number one, they act as highly specific catalysts, greatly speeding up the chemical reactions which would otherwise be hopelessly slow. Number two, they provide mechanism whereby individual chemical reactions can be controlled. The available quantity of enzymes determining the rate and corresponding the reaction can be there. So we can assess it and we can accordingly have an understanding of the same. Well, the sequence of events can be repeated very rapidly. The action of enzymes, catalase, provides the dramatic example. Catalase is found in the body tissues of many animals and plants and breaks down into molecules of hydrogen, provide H2 and O2, releasing oxygen gas and water. The mechanism of enzymes action described above is known as the lock and key hypothesis, but this concept is helpful in only up to a point. and it certainly makes its point as we move along.